have a problem. I found this cool article and I'm like, oh, let's just tell people about it. And it ended up making a video where I tell people like way too much about it. So if you want that way too much about it version, stay watching. But if you just want the quick and dirty, here you go. So basically these scientists, they wanted to see if they could make bacteria only use 19 of the 20 canonical amino acids. And so these are the building blocks of proteins, and there are 20 of them, and they've got these different properties, and then you have different proteins, have different sequences of them in different orders, and then that is then going to influence how the protein folds and thus how it functions. And the relationship between fold and function is one of the key things that's explored in this paper, although all of the headlines and things are kind of like, oh, these bacteria are now using only 19 amino acids instead of 20. The real interest for me is kind of, well, how can we learn about protein structure-function relationships by trying to design these bacteria to only use 19 instead of 20? And the whole 19 instead of 20 thing is a bit misleading. So basically, they're only replacing them in the bacterial ribosome, and I say like only because that is only one of the kind of like protein complexes within the cell, but it's a super duper complex one, so it's a big beast. But they're only kind of replacing the amino acids, so using it only 19 amino acids with the ribosome, not using 19 amino acids throughout the entire genome, which would be an even bigger feat, which we'll get into. Not to be a kill sport, but they actually only replace them all kind of at once in part of the ribosome. So the ribosome has like a large subunit and a small subunit. And the proteins in the small subunit, basically the instructions for making those proteins are nearby one another, and so they could kind of replace them all as this one big piece. And in this way, they were able to replace 21 of the proteins at the same time, but that ribosome has like 50-something proteins, and so although they could individually change each of those proteins and still get a functional bacterium, they were able to design proteins that they could basically make isoleucine-less versions of each of the individual proteins of the ribosome. They only actually tried combining 21 of those in the ones because they were kind of like conveniently located next to one another in the genome. And if they want to, I guess the next step would then be to try to do all of the ribosome, because right now they're really only doing part of the ribosome, although they showed that they could replace one by one each of the different proteins with the version that didn't have isoleucine. Unless I'm reading things wrong, then they actually only replace like all in one for a part of the ribosome. And so in most of the news things and like the summaries and everything, it kind of glosses over that. I mean, it's still a huge accomplishment, but they weren't able to do, or they didn't try to do, they didn't report that they did the swappage of all of the isoleucines in the ribosome at once. So it's not an isoleucine-less ribosome, but it's a partially isoleucine-less ribosome. And then each of the individual parts were able to be made isoleucine-less, but once you put those in combination, who the heck knows? This is kind of showing a proof of principle that you could potentially expand things further, maybe even then open up space for adding in additional amino acids kind of like in its place. But not in its place like in place. Instead, what they need to do is they need to figure out how can we maintain the original proteins with having different amino acids where there was that one that we took out, and then we can kind of incorporate other amino acids in other places elsewhere. Because now we have freed up concordant space in the genetic information that encodes for that specific amino acid. So which of these amino acids are we going to ditch? Ditching any of the amino acids for biochemists is kind of like ditching Pluto as a planet was. I don't know about you, but that hurt, even though I don't really care about space stuff very much. But ditching isoleucine, I'm sorry, isoleucine, I love you, but you gotta go. And so they're taking out isoleucine in these proteins, and then they're trying to figure out, okay, well, what do we put in its place? And so one way is to kind of just brute force test it. Okay, let's change it to every other thing we can. And in some cases, they actually had to resort to this for some of the proteins because their modeling tools were not figuring out what to put in its place. But they did have these modeling tools that were really successful in trying to figure out which amino acids they could replace. they could like put in the place of the isoleucine. And so they tried a bunch of different software programs, and they found different results with different programs. And these different programs were stumbling across things that like evolution wouldn't be able to stumble across, because it kind of like has this linear path, whereas here they're like, okay, let's just take things and just like, okay, machine, now take all of this baseline information about what we know about protein sequence structure relationships, about what we know about these protein structures, about what we know about their sequences, about what we know about the sequence of the same protein in different species. Let's take all that information together, use that information to now try to design proteins that still have this shape and still carry out this function, but have a different sequence, a sequence that doesn't use the isoleucine. And so they test out a bunch of different like machine learning tools. Some of them are based on sequence, some of them are based on evolution, some of them are based on sequence to structure, some are structure to sequence, all these various different tools that they use. And ultimately they're able to kind of redesign the bacterial ribosome to not use isoleucine, even though it still then places isoleucine in place for other proteins. Interesting too was, like, I didn't really think about this until later, after I finished recording the video. I mean, like, I thought about it earlier, but then forgot it when I was recording the video because I just got so into things. But another interesting thing to look into potentially is, okay, what effects does that have on the expression of the genes that these bacteria use in order to make isoleucine? So did they like downregulate making isoleucine, making genes, because now they're not using as much isoleucine? Except in this case, they are still using isoleucine throughout the rest of the genome. In the future, however, they could expand to use to not use isoleucine in those other places, but that's a lot of changes that they need to make inside of the cell. Speaking of changes, they focus on changes at the amino acid level. A key thing for the future would be also to look at the DNA and the RNA levels more and try to take into account the fact that there's a lot of regulatory information that's encoded not at the level of the amino acids, but actually at the level of the DNA and the RNA, that if you're only focused on the amino acids, you might be missing stuff like important regulatory information about how different genes are kind of like functionally transcribed and translated and all these things. And so that's kind of a future direction. For now, the hype is that they're using 19 amino acids instead of 20, whereas like all the organisms we know of use at least 20. So there are some that use more than 20, but not ones that use fewer than 20. And so here they're showing, yeah, we can use fewer than 20, but only in the ribosome at this point in time. And again, like the ribosome is a super complex thing. And so in bacteria, it's made up of like 50-something different individual subunits, so these different proteins as well as lots of RNA and this big old complex. And so if you can show that you can change isoleucine in that to other amino acids, then you can likely do it in a lot of other genes, but that's a lot of work to actually go and change all of them in all of the other genes. And as you'll see in this paper, when they try to do just like simple swaps of like, hey, isoleucine looks a lot like valine, let's just swap it for valine, sometimes that works, but a lot of time it doesn't work. And then they had to go resort to these other tools. Sometimes those other prediction tools didn't even work, and they had to resort to that brute force, like, okay, change, and then be allowing the bacteria to kind of like fight it out, see who survived, and then take those into account. The other thing that is kind of like challenge that they found was that like in isolation, yeah, these proteins might be functional, and even in the like when you're doing it, making these changes in the context of the bacteria, you change one of the proteins, fine. But now you change this protein and this protein. Well, this protein actually was kind of like interacting with this other protein, and then now you have these problems because you don't just have the proteins by themselves, you actually have the proteins in the context of other proteins. And so that information then can be incorporated further into further computational tools in the future. So I think that in the future wise, like things would be to kind of incorporate more of that protein-protein interaction information into the structural modeling, as well as to incorporate the DNA and the RNA level stuff. This is a really cool start though, and I really enjoyed kind of being able to learn more about machine learning, more about these prediction tools, what goes into these prediction tools, where they have different strengths and weaknesses, how you can, and sometimes they don't work, and how you kind of have to combine that with experimental methods, both to validate as well as to come up with solutions that the software can't come up with. And also then to kind of look at the solutions the software does come up with and see what does that tell us about the relationship between a protein structure and its function. And so now that I've kind of like geeked out in this big picture view. So yeah, we took away isoleucine in the ribosome, and we were able to replace it with different amino acids that we were able to predict using these computational tools, as well as able to just like brute force for the ones that would defy prediction. Speaking of defying prediction, our predictions actually defied what we would predict based on our minds, and based on evolutionary constraints, but they were able to come up with these cool solutions using data, though, from experimentally determined data, and then kind of like the iteration between the experimental and the software is kind of like the interface of where things are at this point in time and like moving forward and things. And so you still need the experimental, but here they're doing experimental at like a much bigger scale with a lot of automation and things. And so future work too would be then to kind of like look into these individual changes that they make at more of the experimental level and see kind of how can we learn from the changes that were made in order to help us design other proteins in the future. And so there's definitely limitations, there's definitely strengths, but this is kind of the paper. And so if you want to deep dive, stay tuned. If not, I'm sorry, I just really geeked out about it. And yeah, this is one of the reasons too where I love being the bumbling biochemist and kind of having control over my content and everything, is that I just make what makes me happy and what I think will help people. And so this is one of those ones that just kind of more is because it makes me happy, and there's got to be somewhere out there that could really geek out over this paper with me. And but there's no one like nearby to geek out on this paper with me, and so thanks for geeking out on this paper with me. And if you don't want to, like, don't feel bad. You might want to stay for at least the first few minutes or so where I kind of talk about my strategy for approaching a paper, and then I though go real deep into this paper. So I've already done a deep read of this paper, but I'm going to take you through it, pretending like I was kind of just opening this and showing you my thought process and my reading process as I went through it. And so when I open the article that I want to deep read, there's a few different things that I'm going to do. The first thing is just to kind of scroll through the article and get a sense about what's in the article. And so I want to go and I'm going to scroll through. I'm going to look at the various figures, see what types of experiments they use, and see what I have to look forward to as I read this paper. And so I want to kind of see, okay, well, these are the figures that I'm going to encounter. And then I'm also going to open the figures in the figure viewer window. So I have a separate tab open where I have the figures in bigger view so that I can view them more easily alongside the text and don't have to keep scrolling back and forth. So depending on the journal, they'll have the figure viewer in like different ways, and sometimes there's not a figure viewer. But with science, if you come over here, you can click on figures and it'll pop up a side panel. And if you open in viewer, now you get this larger one. And so I like to do this in a separate tab, and then I just kind of keep it open. I also want to have a tab where I have the methods, so I can go back and forth to the methods. And so depending on the paper, the methods will be in different places. And so the methods are in the bottom of this article, so like at the end of this article. And so I just opened a separate tab where I have the methods section set here, so I can go back and forth between the article and the methods without having to scroll back and forth. Also, at the end of a paper, you often find supplementary materials. One of the first things that I like to do when I'm reading a paper is after I scroll through the paper, I look and see if there's supplementary materials, and if so, I download them. So you can see that here there are some tables. Often I won't really look to the tables much, but there's also this PDF file with supplemental figures, and so you can see figures S1 to S17. If I press download, I get this PDF. I open it in another tab, and now I also have the supplemental figures that I can refer to as I'm reading the paper. I like to start by scrolling through those supplemental figures and kind of get a sense about what's in them, similarly to how I scrolled through the article in order to get a sense of what was in the article figures. Often in the supplemental, there's going to be kind of models, like graphical abstract type of things, as well as there might be additional methods and there might be kind of schematics that are going to help you understand things, such as the methods that were used. And here they're also telling you what's in those tables if you wanted to go and look at the tables, because often the tables are kind of not going to be named very, hopefully they're just going to be S1, S2, S3 or whatever, and then here it's kind of telling you what's in each of those tables. And then again, kind of I look and see what's in these supplemental methods, and you can see like a figure like this is going to help me understand some of the methods. And so I want to have the supplemental materials open in a separate tab. I also want to have just kind of like a search engine open so that I can look up terms that I don't know, techniques that I don't know. For example, this paper uses a technique called Lambda-Red. I hadn't heard of it before, and so I did some Googling on what that technique is. And I also kind of use the references from the paper in order to find more information about various techniques that the paper used that I didn't know about. But speaking of that paper, let's get back to it and let's go to the start and go all the way through it. We're going to go in depth. So this is much more in depth than you would go often when you're reading a paper. And I'm going to be kind of like presenting it, not really presenting it in the way that I would in a journal club, but really walking you through all the different details of the paper to the best of my understanding. And so it uses some techniques that I'm very familiar with, others that I'm not so familiar with. It uses some AI, machine learning type of things that I'm not very familiar with. In terms of the deep workings of it, I'm not sure how they work, but I understand kind of some of the logic that goes into them, at least I think so. And I'm going to explain things to the best of my knowledge, and if you find any errors in my explanations, please do feel free to let me know so that I can correct them, because I want to make sure that I'm explaining things correctly, and I want to make sure that I understand things correctly. So let's dive in. In addition to scrolling through the paper to see what's in it, I also like to just kind of like look at where the paper is coming from. And so if I go to authors and affiliations, I can see kind of where these different authors are from. And so the authors at the end, the ones with like the envelope, these are going to be the corresponding authors. And so these are going to be kind of the head of the labs where the research was taking place. And so you can see that we have work being done at Columbia. We have work being done at MIT, Harvard, various places like this. And the first author is going to be the one that is kind of typically doing the most of the work. And you can see too that there's kind of this symbol here that is indicating that these people, there are like multiple first authors that contributed equally. And you can see too that sometimes it'll tell you what they contributed to the article. But speaking of the article, let's dive in. So here they're starting with an editor summary, and so some papers will have this and some won't. And this is going to then tell you kind of a broad summary for this is more for an outsider perspective, a more general audience type of view about the importance of the work. And the editors might say like, okay, this is kind of like why we chose to publish this because we think it's this cool thing and blah, blah, blah. Then you have the actual abstract from the authors that were kind of summarizing their work. And so here they're introducing their work, the rationale, the results. Abstracts are going to be formatted differently in different journals. And so here you have kind of what's called a structured abstract, whereas typically an abstract is just going to be like a one paragraph thing. Here's the structured abstract where it kind of breaks things down into introduction, rationale, and results, and a mini version of kind of, and conclusion, and a kind of mini version of the actual paper. So this is kind of unusual to have this structured abstract like this. A lot of times you're just going to have a single paragraph that's kind of an overview of the work. This is what we call a graphical abstract, and so here is kind of a graphical overview of the project and of the paper before you get the actual paper. And so here's this more like typical abstract that you would see where you have this single paragraph explanation of the paper. And all of this is before you actually get to the paper itself. And so let's get to that paper itself. Before we dive into the paper, I like to look at the graphical abstract if there is one, and that kind of helps set the scene. So we can see that there is going to be kind of, they're showing that we have this amino acid alphabet, which has these 20 letters, and they're going to try to remove the isoleucine, this I, and replace it with other amino acids in order to recode an E. coli cell to only use 19 amino acids. But they're only going to do it in the ribosome, as we'll see. How are they going to do this replacement? Well, there are the methods that they use to physically do this, and then there are the methods that they use to design the changes that they're going to physically make. And so they're going to be using this kind of strategy where they're going to be using a variety of different software tools, some different AI tools that are going to rely on using the sequence as well as predicted structures and known structures in order to predict which changes would be least disruptive to the functioning of the bacteria. They're going to do this in the proteins that make up the E. coli ribosome, in addition to all of the RNA and stuff. And so you can see that they're going to basically be replacing the isoleucines in these different proteins. When you see this delta sign, that means there's like missing or without, and so this is without isoleucine, and so there's going to be a change in the ribosome at 382 positions where they're replacing the isoleucine with something else. They're going to be using that generative modeling software in order to kind of predict what changes to make. They're going to make those changes using genome engineering, and then they're going to actually test whether the E. coli are still able to survive and whether they're grown normally. And so they call this like DBT, design, build, test. They're designing it. They're then using the genome engineering to make it, and then they're going to test to see whether the bacteria are still able to survive. And so this is what we're kind of going to be looking for throughout the paper, so keep this framework in mind. And now let's dive in. Okay, so the kind of rationale again is that we know that all living organisms have at least 20 canonical amino acids. And we know that evolutionary speaking, you had to start with fewer than 20 in order to get to 20. So how did you have functional ribosomes with less than 20? And can we recode the genome to use less than 20? This would have benefits such as freeing up extra space in the genome for us to then take over those codons that were kind of typically in isoleucine. And can we use the codons that were typically specifying isoleucine in order to now specify some sort of unnatural amino acid? So kind of replace isoleucine, not in the positions it normally was, but in terms of introducing its codons elsewhere in order to introduce other amino acids elsewhere. So there's a bunch of cool synthetic biology applications that you could think of coming from this, but the first step is to kind of see whether it's even possible and to see the power and limitations of the predictive software tools. Often in the introduction, they're going to introduce some of the acronyms that they're going to be using throughout the paper. And so it's important that if you come across an acronym that you're not sure of, the first thing you want to do is to kind of look and see whether it was introduced in the paper itself. So often you might have a tendency to just go straight to Google and Google the acronym, but the acronym probably stands for a lot of different things, potentially even a lot of different things in the scientific context. And so the first thing that you want to do is actually look to the paper itself and see where they introduced the acronym. And so just do a Control F, and then you should be able to find it. For example, the CAAs, it stands for canonical amino acids. And so maybe I was reading through the paper and I kind of skimmed through the intro and I come across that term CAA, and I'm not sure what that means. I'm just going to Command F, and I'm just going to go CAA. And oh, it's telling me that stands for canonical amino acids. Okay, cool. And it uses it 17 times in the paper, so I'm definitely going to want to make sure that I know what that abbreviation stands for. And so they're going to be trying to remove one of these canonical amino acids, isoleucine, from the ribosome and replace it with other canonical amino acids. And they talk about canonical amino acids because there are also some kind of additional amino acids in some species that we're not going to go into here. Another acronym that we'll see is LUCA, so the last universal common ancestor. So basically, the last ancestor that we have shared genetically had, with like all organisms, had 20 amino acids, these 20 canonical amino acids, and there aren't organisms that we know of that have actually lost some of those amino acids. So there are some organisms that have more than 20, but not ones that have fewer than 20. But synthetic biology might now offer us the way to kind of design organisms so that they have less than 20. And if we can do this, it's going to kind of tell us a lot about evolution, about the software tools, advance these software tools, advance our knowledge of proteins, and kind of know our limitations with these sorts of things. Okay, so here's another acronym that we're going to come across. It's BCAAs. So BCAAs are the branched chain amino acids. And so if we look at the structure of amino acids, we'll see that they have different properties. And some of these have what we call branched chains. And so these ones are going to be our valine, our leucine, our isoleucine, and our threonine. And so you can see that they kind of have these branches in these side chains. So you have these 20 amino acids, these 20 canonical amino acids, and they have these kind of generic backbones that allow them to link together, as well as these unique side chains, or R groups, that stick out. And because they have unique properties, they're then going to influence how the protein folds in order to best accommodate those unique properties. This is isoleucine, the one that we're going to be trying to replace. And we can see that it's got this branching network in its side chain. And you also see that its side chain is made up of carbons and hydrogens, as opposed to containing things like oxygens and nitrogens. By just having carbons and hydrogens, it's what we call nonpolar. And so basically carbon and hydrogen, they share the electrons fairly evenly. And so electrons are negatively charged, and these atoms kind of share them equally. And so this is going to and that balances out with their protons, and so then this is going to be kind of a blah side chain in terms of it's not going to be very appealing to water and things, so it's going to be hydrophobic, and we call these like nonpolar. And the branched nature is also going to make it so that it's kind of hard to rotate around. And so if we think about the branched chain amino acids, we've got our valine, our leucine, our isoleucine, and our threonine. But threonine, it has this oxygen in its side chain, so it's polar. Water likes to hang out with it. It's hydrophilic, as opposed to this leucine and the valine. And so leucine and valine are kind of the closest similar amino acids to isoleucine. And so we'll see that one of their strategies is just to actually go and take every place that there was an isoleucine and change it to a leucine or change it to a valine. So some of the abbreviations that you're going to want to get familiar with are for isoleucine, we have I, leucine, we have L, and valine, we have V. One of the most important things to do if you want to be a biochemist is to actually memorize the abbreviations for the amino acids, because these are going to show up. And we're going to see that we're going to have a lot of substitutions of I's to L's and of I's to V's. But we'll also see that that kind of just strategy of replacing them by the ones that look similar isn't always the most ideal solution, and just doing that brutally, just replacing them by leucine or valine, often is going to lead to dysfunctional proteins. However, by using these generative AI tools, they're actually able to find alternative solutions to making functional proteins, and some of these solutions are going to kind of be kind of out there and stuff that you wouldn't necessarily expect, because these tools don't have to follow kind of your traditional evolutionary pathways. And so we'll see some kind of cool solutions to protein folding and that allow for the making of functional proteins. So in the introduction, the scientists are going to kind of lay out where the field stands now, why this work would be interesting, and what evidence is there that this work is even feasible. So here they're talking about the feasibility of the experiment. So as we were just talking about, we have these branched chain amino acids, and they have overlapping hydrophobic and steric profiles. Okay, let's break that down. We talked about hydrophobicity, so these branched chain amino acids are nonpolar. They don't really like to hang out with water, or water, more accurately, water doesn't like to hang out with them. And so typically these amino acids are going to be found kind of buried in the interior of a protein. As for steric profiles, well, sterics, this is referring to kind of the space that they take up in 3D. And so we can see that these branched chain amino acids were kind of similar in size and similar in the distribution of their bulk, meaning that if we were to replace them with one another, it wouldn't cause too much disruption in the neighboring amino acids, the neighboring structure of the protein. As opposed to replacing something like isoleucine with something like tryptophan, which is a lot bigger, that could kind of have more steric influence, Or if you were to replace it with something like glycine, that would be very small, and that would allow for more flexibility. And so the steric nature, they're kind of referring to how much space they take up in 3D and how that space is kind of taken up in terms of where the bulk of the structure lies. Okay, so they say that we have these branched chain amino acids, and they're highly redundant in terms of their shape and their properties of hydrophobicity. Computational tools have suggested that you only need 9 to 12 amino acids to make almost all protein folds. And so we're thinking about amino acids in isolation, but really amino acids are kind of only one layer of the structure of a protein, and then how those amino acids are interacting with one another generates distinct folds in the forms of like secondary structures, things like our sheets and our beta pleated sheets, our alpha helices, that then give you larger folds that make up the tertiary structure, the bigger 3D structure of the protein. And then, especially in the case of a protein like a ribosome, well, that ribosome is actually made up of lots of different proteins, and then how they interact with one another and how they interact with the RNA to make the functional ribosome, that gives you what we call the quaternary structure. All of that is built off of information from that primary sequence, that sequence of amino acids. However, you have these multiple layers that are deriving from that. And so they can say here that the sequence information of those amino acids is really important, but you can kind of change the sequence and still get similar folds of the protein. So similar 3D shapes, despite having differences in the sequences at the amino acid level. They talk about how the codon, the genetic code is kind of plastic, and so there's a lot of flexibility in it. And so in some organisms, they've actually used some of their codons to code for other different amino acids. And they talk about how there's actually a lot of natural kind of mistake making in the making of proteins. And so a typical translational error rate of about 10 to the -4th by the ribosome leads to about 8% of cellular proteins containing at least one misincorporated residue. So basically, the ribosome makes mistakes sometimes naturally, and the cells are still functional. And so this offers some evidence that we can purposely make changes, and the cells will still be functional. Now they're talking about kind of evolutionary stuff about which amino acids likely arose earlier. And we're just going to kind of scroll through that. They talk about some of the other synthetic biology work that's been done. Most of this has just been kind of reassigning codons so that instead of telling the ribosome, hey, add this amino acid, they say, hey, add this amino acid, and things like this. And so, again, this is kind of more of the rationale, the background. And now they're going to tell you about some of the tools that they're going to be using and why this is now the time to do this experiment. And so they're going to be using a variety of different computational protein modeling tools. And they're going to be using it as part of this DBT framework, this design, build, test framework. And they're doing it through an iterative design, build, test framework. And so basically they're iterative, they're going to be doing it in cycles, and in each cycle they're kind of improving and then improving and then improving to try to get the best. And they're going to be using a variety of different software tools in order to do this. And as we'll see, they'll have to use different tools in order to kind of work with different proteins. And some proteins will work better with some tools and some with other tools. And so they're trying to figure out kind of the strengths and limitations of these different tools, as well as their kind of more overall project goals. They're also going to kind of combine the strengths of these different tools, and also some cases just brute force in order to make these functional ribosome without any isoleucine. And so these are some of the tools that they're going to be using, and we'll talk more about these as we get into them. As this mentions, however, these tools have not been used for this before because this is kind of novel research. And so normally these tools can be used in order to predict structures of proteins and as well as to predict sequences from structures and things like this. But all of those are still using those 20 canonical amino acids. And here they're saying, okay, well, now we're going to take these tools and we're going to tell it, hey, don't use isoleucine, and see if the tools can still do something, can still work as normal. Or not as normal, but they can still work. Okay, so we're getting into the meat of the project now. And so they're going to be using the model bacterium Escherichia coli, so E. coli. When we talk about a model organism, this is kind of a organism that scientists have done a lot of work on. They know a lot about it. There's a lot of tools for working on it, and so it makes it kind of easier to work with. And rather than trying to do this with some sort of organism that scientists don't know much about, there's not many tools. With this E. coli, we have a lot of information about it already that is then going to help the scientists in their experiments. And it will help other scientists be able to kind of replicate the work in order to verify it or to build upon it. So they're going to be using different protein language models, so PLMs. This is another one of those acronyms that you're going to want to get familiar with. And so when you're reading a paper, you might want to actually be kind of writing down terms that you don't know of, or acronyms and kind of their meanings. And remember too, just Command F if you have an acronym that you don't know what stands for. You want to look for it in the paper, so either in the paper itself or sometimes in like figure legends and things, before trying to just go Google and see what it means. So they're going to be using these protein language models, these PLMs, as well as structure-based predictors. And so we'll talk more about this later, but they're going to be actually incorporating 3D structural information in addition to the sequence information in order to predict structures from sequence and sequence from structure, and then allow flexibility in those predictions in order to find kind of more novel solutions to folds and sequences and their kind of interactions. And so we'll get into that in a second. So they're going to recode essential genes. So essential means that basically if you take them out, the cells die. And so these cells need these essential proteins in order to live. If they make changes to those essential proteins that are functional, that allow for the proteins to still be functional, the bacteria will still survive, they'll still grow. If the changes make the protein non-functional, then the bacteria will die. And if the changes kind of make it so that the protein is somewhat functional but not ideal, then you'll have reduced fitness. And so they're going to be monitoring the growth of these bacteria in order to figure out the kind of fitness costs of various mutations that they make. And so we'll look in a second at the methods about how they're going to be actually measuring that. And so in this paper, they kind of set this threshold of they want to have greater than 90% fitness compared to the wild type proteins. And so they're saying, okay, these grow at least 90% as well as the normal unmodified versions of the proteins. So cells with the unmodified versions, what we call the wild type proteins. So when you see wild type, when you see WT, it's referring to wild type, and that's referring to the unmodified version. They're going to do this in isolation for each of the different proteins, and then they're going to combine those proteins, the design proteins, together and in a native operon. And so basically what they're saying is that native in place in the bacteria, they're going to be putting in these new versions of the genes to make new versions of the proteins that don't use isoleucine. And they're going to do this as part of an operon. And so an operon is basically where you have a bunch of genes that are made together, so they're expressed together. And so a lot of the different proteins of the bacterial ribosome are made kind of at once. So the ribosome goes and it makes these all at the same time as one continuous piece of DNA called an operon. And so they're able to kind of replace a lot of the genes as this one kind of chunk in place where it would normally be in the chromosome. And so we call that a native operon. And so you can see here that they're kind of taking this sequence that had these instructions for making all of the usual proteins for that ribosome, or at least a lot of them, and then they're going to go and they're actually going to put in a version that has the different, the changes to make the new proteins. And we'll go into how they're going to do this in a minute. but that's then going to allow them to do things in place and have the least amount of disruption, and to actually be kind of re-engineering the genome. So they call this like recombinering, where they're recombining pieces of DNA using this recombinase system we'll talk about, in order to make these bacteria have the instructions for making these isoleucine-less ribosomes. And then they're able to test whether those cells are actually viable. And they're going to do this using AI-guided tools. One thing, too, though, to note the limitation is that they are just doing this in the ribosome. And so the ribosome is this super complex piece of machinery. It's got a lot of different proteins. And these proteins have to interact with one another and with RNA. So it's a very complex protein. It's a very complex system. And you would imagine that would be a lot easier for other proteins in the organism. But at this step of the process, they're just doing it with the ribosome. And so although it's kind of talking about a 19 amino acid alphabet species or whatever, they're only doing replacement in the ribosome at this point in time. But you can definitely imagine that they're going to be wanting to do it further. One of the limitations, however, is then you have to go and change all of those different genes in the bacteria that have those isoleucines, and then you're going to actually have to change each one individually and make sure they're all changed individually. So there's a lot of work that needs to be done, especially because, as we'll see, although they kind of started by changing one protein at a time, they found that even when they found optimal designs for those individual proteins, when they tried combining them together, it didn't always work. And you have a ton of different combinations to try, because their models, they don't just give them one possible solution. They give them a lot of different kind of versions of the protein, all of which might be good, some of which might work better with other versions. And so you get all these combinatorics that can make things very, very complicated. And then you get to thinking about all of the kind of how do we do this in the lab, and how do we do this feasibly, and then all these like big machines doing things. And so it's not an easy task, but this is kind of a proof of concept showing that you can do this for the ribosome, which is really, really cool. And so let's go through this paper in more detail. So again, kind of my workflow. I'm going to go through this paper. Often what I do is I actually look at the figures, and I kind of try to interpret the figures without having read the text yet. But I'm going to kind of do things side by side with you to help make things easier for you to interpret, and so I don't jump ahead of myself. I'm going to have the figures open in another tab as well, so I can quickly refer to them. And so here's just kind of that graphical overview that we were looking at. And now let's go. This is going to be our figure 1. And so I like to kind of look at the figures and see what's in the figures, and then I can refer to these figures as I'm reading, and I have the supplemental figures I can refer to as well, because often these supplemental figures are going to be kind of referred to in passing at the same time as they're referring to the larger, kind of like the main figures of the paper. So the first thing that they're going to want to do is they're going to try to identify which amino acid that they want to change. So remember, CAA stands for canonical amino acid, and that's one of those 20 main amino acids that we were talking about. And so one of these 20 amino acids, which one are they going to try to replace? So they know that going into this, they want to try to replace one of them, but which one? There are these 20. Which one is the one that they want to replace? And so here they're going to be computationally analyzing feasible replacement strategies based on evolutionary sequence conservation and PLMs. Whew. Okay, so what they're going to do is they're going to look at the sequences of proteins that have been kind of conserved over evolution. If there is an amino acid that is kind of used a lot in the same places in the same proteins in different organisms, and so different protein orthologs, so basically different versions of these proteins in different organisms, if these proteins have kept that amino acid, so if all those proteins still have an isoleucine in that place and things in the protein, then that's likely important. Whereas if the amino acid in that position is kind of changing a lot, it's different in these different orthologs, these different versions, then it's likely that the actual identity of that amino acid, so which of those 20 it was, is less important. And so they're going to look to try to find one of the amino acids that is kind of in less of those conserved positions where there's kind of this amino acid isn't as consistently used in these different proteins, in these different organisms, that would be a good candidate for something that we might be able to change, or that they might be able to change. And so they're going to look to evolutionary sequence conservation in order to kind of try to figure out which of these amino acids would be the best to try to substitute. They're going to be doing this in E. coli, as we talked about, and the specific strain is going to be this K12. And they're going to be using this as this model cell because it's the most studied. It has a genome, and that genome is well known about, that has actually been previously recoded and synthesized de novo, so from scratch. So basically, previous scientists have actually been able to kind of make this genome from scratch, and so you can go, and if you want to read that paper, you click on there, it'll take you to the references where you can see, okay, this is going to then be a paper that I'm able to read, and you can even go open it in another tab. Open it in another tab, save it to your Zotero to read later and things. Sometimes it's helpful to go and look kind of as you pause and go through and look at different things, but other times it kind of takes you on rabbit holes, and so be careful. So they're going to call this effort EC19. So E. coli that uses 19 amino acids. And so keep this acronym in mind too. This one's going to come up a lot. So we know what they want to try to do. Now let's see how they did it and what they found. So how did they go about this? So now we're getting into some sort of methods. So the actual methods section is going to be at the very end of this paper. And so again, I have a tab open where I have it starting at the methods so that I can actually go and scroll through and look in more detail at these methods as they're being introduced in the paper. Sometimes the methods are going to come before the results. And so that's one of the reasons too where I like to scroll through the paper and kind of get a sense about the layout of the paper and about where the different results are going to be, where methods going to be, and then kind of open up tabs to different important sections so that I don't have to keep scrolling back and forth. So for each of the canonical amino acids, they calculated a global conservation score from a multiple sequence alignment, or MSA, of each of the E. coli proteins and its orthologs. Okay, so let's translate this. We have each of the 20 canonical amino acids. They're going to calculate a global conservation score from a multiple sequence alignment. So if we go to the materials and methods, we go to this section where it's telling us about this MSA construction and this conservation analysis. And so basically, they retrieved the gene orthologs from the OrthoDB database. So they went to this database that has all of these information about this protein, different orthologs of the protein, so different versions of it in different species. They restricted it to just comparing bacteria, and a lot of these kind of have the same sequence, or they're kind of like versions from different strains of the same bacteria and stuff. And so they kind of used tools in order to filter things out so there was less redundancy and they had more variation in their sequences. Because their goal is to kind of see if variation is possible, not just how much of variation there is. And then they talk about some sort of like technical tools, some software things, and so we're not going to go into all those technical details. But what we do want to look at here is this residue conservation analysis. And if you were just reading this paper for pleasure reading, like, you might not be going into depth into all these various things, but I just want to geek out and kind of show you kind of how these things work at a deeper level, because this is the kind of information, these kind of conversations that I feel like are very rare to find, but can also be a lot of fun and can be hard, however, if you're unfamiliar, to kind of even know where to start. So what they're saying here is how they're kind of measuring and determining the conservation level. And so they're saying, okay, they're saying if we'll define an amino acid as conserved at a position I, if its relative frequency is 95% or more. And so if 95% or more of the sequences of the protein orthologs, so the versions of the protein, have that amino acid there, basically we'll call this conserved. And then they go and they say for all of the conserved positions for each amino acid type across each of the essential gene sequences, so all those ones where the genes where if you mess it up, the bacteria can't live. And then they're kind of like normalizing that by the total occurrences of that amino acid in the sequences. Then that would kind of correct for composition bias. And so if that amino acid kind of occurs a lot in those proteins, they want to just kind of like normally, then the information is kind of less valuable, and so they need to be able to normalize and correct for that by like normalizing to the total occurrences of the amino acid in the sequences. Okay, so that's kind of like the basic idea, is that they line up all these sequences and they look at each position and see, okay, well, is this amino acid conserved at the position? And how often is this amino acid conserved versus other amino acids? And the results of this are going to be shown in this figure 1C. And so here we're seeing the global residue conservation across all orthologs. And so when you're looking at a figure, some things that you want to do, you want to look at the axes, and you want to look at kind of the figure legend information, the keys and things. And then if we look at the legend, we can see that C is the global conservation score of each of the 20 amino acids based on those MSAs, so those multiple sequence alignments, of the E. coli K12 essential and non-essential genes with their orthologs. And so they take the essential genes, the light, the non-essential genes, the dark, for each E. coli protein, and how well is that conserved in the protein in other species. They find that cysteines are kind of the most likely to be conserved, whereas valines were the least likely to be conserved, followed by isoleucine. And so the branched chain amino acids, and so often we don't really think about threonine as a branched chain amino acid because although it is kind of branched chain, it's also polar, so it is less similar to the leucine, the isoleucine, and the valine, and it's kind of processed slightly differently in the body. And so they're kind of just going to be considering valine, isoleucine, and leucine. And you can see that valine and isoleucine are kind of the least conserved ones, which makes it likely that you might be able to kind of substitute these more readily than substituting something like a cysteine or a glycine or a tryptophan. Now, you might be wondering, why can't you substitute cysteine, glycine, or tryptophan? Well, if we look at the structures of these, we can see that, well, tryptophan is just kind of like this huge bulky thing, and so now we get back to thinking about kind of sterics, so how they take up space, as well as tryptophan is able to form some interactions, some hydrogen bonding and things through this nitrogen. Glycine and cysteine, well, these are two of our kind of weirdos, and so glycine is basically its side chain is just a hydrogen, so it's really, really small and it's flexible, and so it's going to make regions of the protein that incorporated glycine really flexible, really loosey-goosey. Cysteine is going to be able to form disulfide crosslinks, so it's got this sulfur at its end, this sulfhydryl group, this thiol group that's able to form covalent crosslinks, or basically strong links between regions of a protein and regions of one protein and another protein. And those are kind of functions that aren't carried out by any of these other amino acids. And so if you change a cysteine, or if you change a glycine, or if you change a tryptophan, that would have kind of like big consequences. Then you have proline, and proline is another one of those weirdos where it's not like the others, and so it twists back on itself. It kind of constricts the region of the protein. And so it too would be one that you probably wouldn't want to go messing around with. And they agree, and they also say, again, like the isoleucine and valine have much biochemical similarity, typically reside in the buried hydrophobic cores of proteins away from active sites. And they're now directing us to the supplemental, where we see this kind of like figure where they're showing us, yeah, isoleucine and valine, leucine, these look pretty similar. And then they're going to use some computational tools to kind of justify things further. And so they're using this tool called ESM2 PLM. So they're using this like artificial intelligence or this multiple sequence alignment on those E. coli essential proteins. Then they're going to be doing like pairwise replacements for one canonical amino acid to another to produce a blossom-like substitution matrix. So what they're trying to do is kind of look and see, okay, well, we know that these are less conserved, but what amino acid are they typically substituted for? And so they see that kind of this isoleucine to valine substitution is going to be one of the most prevalent ones, as predicted by both of those software tools. So both the ESM as well as the MSA are both going to be kind of saying, okay, well, this one is going to be kind of the most tolerated, the most evolutionary frequently encountered one. And so this would be a good one to kind of change. So first pass on isoleucine to a valine might be just kind of a good way in order to test the feasibility of this. And then they're kind of comparing the two tools here and showing that they're similar in terms of the conclusions that they draw. And so here, if we go down here, they're going to kind of tell us a little bit more about how they generated those figures. And so they basically went through and looked to see the probabilities that that amino acid would be substituted for another amino acid based on kind of the predicted values from the ESM2, as well as the actual values that, like the actual amino acid frequencies that they see in the multiple sequence alignment. Okay, so they've got that. Now let's say, okay, so we know that these two methods are kind of converging on showing us the isoleucine to valine, so this I to V, remember those abbreviations, is going to be a kind of good candidate for doing this. Although they do mention that there were some slight differences between the different models, and so that's something that people might want to look into further and kind of distinguish between these different tools. But isoleucine to valine was this kind of most common one. And well, why isoleucine to valine as compared to valine to isoleucine if they want to do this? Well, what this is kind of cool too is they talk about isoleucine is actually more energetically costly to make for the cells versus the isoleucine. And so it's cheaper energetically for the cells to actually make this valine than it would be for it to make isoleucine. And isoleucine occurs less frequently than valine. And so isoleucine to valine seems to be kind of a good starting point if we want to go and introduce these changes. We'll also see that they do some isoleucine to leucine changes. What other evidence do they have for choosing isoleucine? Remember, they're going to be going into like a huge investment of time and energy and all these things, so they want to make really, really sure that they're choosing the optimal amino acid to try to replace. And they're saying that isoleucine oxytrophy can result from the deletion of this gene without causing valine oxytrophy, whereas no valine biosynthetic genes can be deleted without also causing isoleucine oxytrophy. So let's talk about oxytrophy. That is basically saying that if these cells have this mutation where they don't have one of the genes that's needed to make isoleucine, then these cells cannot survive unless you give them isoleucine. So normally these cells can make their own isoleucine. If you take away one of the genes for making isoleucine, these cells cannot survive unless you give them isoleucine already made. However, those cells don't need to have valine added given to them. They can still kind of make their own valine, but they can't make their own isoleucine. But if you take away one of the genes for making valine, now the cells can't survive unless you give them valine and you give them isoleucine. And so it's kind of, if you mess with isoleucine synthesis, you don't necessarily mess with valine synthesis. But if you mess with valine synthesis, then you do mess with isoleucine synthesis. And so messing with isoleucine seems to be would have less of an effect, you would think, than messing with the valine. As a last piece of evidence, they say that cells that have kind of like an effective version of isoleucyl tRNA synthetase. And so basically this is going to be the protein that sticks the isoleucine onto the tRNA to take to the ribosome. If that kind of proofreading ability of it is low, then it can kind of accidentally incorporate a valine, or this amino acid called norvaline, in up to 15% of the time, but those cells can still survive. And so they're saying that the placement of isoleucine can often kind of have errors, and that could still make for functional proteins that allow these bacteria to survive. So overall, we've got this feasibility, proof of concept, all this stuff, and so let's actually see what they can do. Doing this at the whole genome level would be an enormous task. That we say that basically there would involve over 81,000 isoleucine sites that they'd have to change in the genome, and 7,500 of those would be in essential proteins. And so they're directing us to 1D. And so if we look at all the protein coding genes, we have all these genes, and then this little slice of it is the essential ones. Remember, those are the ones that you can't mess with without like killing the cells. And so you see, like, with the isoleucine residues, and so with the isoleucine positions in those, you've got kind of over 81,000 isoleucine encoded for in the genome, and like about 7,500 of those aren't essential genes. And so they're like, okay, we need to find something a smaller piece of the bucket. And so they're going to start by kind of looking at these ribosomal genes and seeing, okay, well, we've got only 52 protein coding genes in our ribosome, and we would only need to make 382 isoleucine swaps in order to make a ribosome that was fully isoleucine-less. And so that's where, like, we'll see 382. 382 is referring to the 382 isoleucines that they replaced in the ribosomes of these bacteria. And as their first kind of go about to do this, they're first, though, going to look at just kind of doing it for the essential genes. Then they're going to change their strategy and do it just for the ribosomal genes. So they're going to start by taking a selection of these essential genes and kind of just starting with brute force. Let's go ahead and let's change each of those isoleucines to a valine and see if things are still functional. So they choose 36 representative essential genes and 3 highly expressed genes. So basically they say, okay, we're going to take 36 of these essential genes. So of these 398 essential genes, let's take 36 of them to try this out on, as well as 3 of genes that were just highly expressed, because if the cells make a lot of it, then it's probably important for the cell, and so if we change it, even if it doesn't kill the cell, it might still have a big fitness cost. And so what they're going to do is they're then going to be replacing all the isoleucine residues with either a valine or a leucine. And then, okay, so they're going to call these genes recoded. And these recoded genes were synthesized de novo, so they're made from scratch. So you can just have, like, companies synthesize these. And they're introduced along with a co-translated antibiotic selectable marker, CAN-R, into E. coli cells to replace or inactivate the wild-type essential genes at their native genomic loci through lambda-red homologous recombination. Okay, so let's go through kind of how this method works and what their strategy is. They're directing us to figure S2 and the materials and methods. So if we go to the supplementary figures, this is one of the figures that we kind of saw, and I said this is going to be one of the methods figures that we might want to look back at later. What their strategy is they're basically going to be taking a target gene. So this is the gene that they want to change. They want to change it in place. And so they could just kind of like stick in a new version of the gene, but then the cells would still have the old version. And so that would confound the results. They could inactivate the old version and then stick in a new version like elsewhere, but then that would also have kind of, you couldn't tell if any changes that you see were because you put in a copy in this artificial way or because you had made the change. So what they want to do is they want to make the change in place with as little other change as possible. In order to make sure that cells actually make the change, or that they only keep cells that make the change, what they do is that they're going to kind of replace the existing gene with the recoded version of the gene, that one that they kind of like synthesize, the one that they make, with those changes in the sequence that are going to lead to the changes in the protein. And also on that piece of DNA that they incorporate, they have this antibiotic resistance gene. And so now the cells are going to kind of replace the normal gene with the new gene, and also with the kind of having this antibiotic resistance gene, so that the cells that make this change are now going to be able to grow if you give them that antibiotic, whereas other cells are going to die. And so you grow them in media, or basically food, that has the antibiotic, and then only the cells that have actually made this swap will be able to survive in the presence of that antibiotic. And so those cells then that didn't make the swap aren't going to be able to survive. And so this is their strategy for kind of replacing things. They're going to be using this homologous recombination strategy. So when we talk about homologous, that's kind of like the sequences. In this sense, the sequences are just kind of very similar. And so the cells are going to say, hey, this looks like this and this looks like this, and then make the swap. But they're only able to make the swap if they have the enzymes, the machinery for making the swap. And that machinery that they're using is kind of this lambda red system. And so wasn't really familiar with lambda red. And so then I looked it up and I found some more information about it. Agene has a nice piece on it. But basically it's this system where these bacteria are kind of engineered to incorporate this machinery from this lambda phage. So from a virus that infects bacteria, they're able to incorporate some of the enzymes of that in order to make it so that you get this swappage more easily. And so it's got these kind of different pieces that are then going to prevent the DNA that you put in these cells from being degraded. They're going to be making the DNA in a way that's going to be easy to swap in, and this sort of thing. And so I don't want to get too distracted by talking about this method, but you can learn more about it if you're interested. And then in the actual methods, they're also going to tell you more of the experimental data about how they did this. And so they're telling you kind of about how they went about making these genes or having them synthesized, putting them in the cells. So they basically use electroporation, and so they use electricity to put these pieces of DNA that they make into the cells. And then they activate this kind of recombination pathway to allow those swaps to be made. They grow it in the presence of the antibiotic so that only the cells that have made the swap are going to be able to have a chance at surviving. But now I need to see, okay, well, did they actually survive? And if they survived, did they have a fitness deficit? And so how are they going to be analyzing the fitness? If we go somewhere in here, fitness measurements. And so basically what they're saying is that they're going to be measuring the growth of these bacteria. So they're going to grow these bacteria in the food, the medium, that is including the antibiotic. And they're going to be doing this in these 96-well plates. They're going to then be doing this in like a plate reader. They're shaking the plates. They're incubating them at 30 degrees Celsius. And then they're kind of measuring the growth of these bacteria at different time points based on the OD600. So basically how cloudy the solution of bacteria is. The more bacteria there are in the solution, the higher the OD600. They're kind of like more cloudy, the less light that can actually get through, the more light that gets scattered and things. So they look over time and see, okay, compared to the unmodified version, how well do these cells grow? And instead of just comparing them to like the totally unmodified, what they're often going to be doing is comparing them to versions that have kind of the replacement of the normal gene with the antibiotic resistance, to just kind of account for any changes that were introduced as a result of having that antibiotic gene, as opposed to changes that come from having the actual difference in the protein, other protein that you care about. And so now they made those changes. They did the I to V or I to L. So they're starting with kind of just the simple changes. So they take an amino acid, they change the isoleucine to a valine, or they change the isoleucine to a leucine. Here they're showing you kind of, they did it in these essential and highly expressed genes. And so they tested 39 of them, and now they're going to measure the relative fitness. And so this is kind of the saying, okay, this is the total number of amino acids in this protein. This is the number of isoleucines in this protein. This is the substitution that they made. So this would be an isoleucine to a valine. And this is the substitution of an isoleucine to a leucine. They're not showing us all of them. They're only showing us some of them. But they're showing us ones that are kind of going to tell us some different things and different things that they recognized. One is that some of these were kind of not able to grow. And so if we look at like this ISPG or this RSPJ, when they made these changes from isoleucine to valine, we don't see any growth. And similarly for these proteins with isoleucine to leucine. We see that some of the changes were well tolerated. Some of the changes had a fitness deficit that came from kind of sequence information at the DNA level and things, and so non-canonical start codon. Some of these had changes that were kind of due to things other than the actual amino acid sequence, but instead due to the codons that were encoding that amino acid. And so it's important to think too, one thing that's going to come up is that you have to think about things at the level of the DNA and at the level of the RNA, and not just at the level of the protein. And so that's what they're indicating here, is that these genes kind of had these non-canonical start codons that made them harder for the cells to make, rather than just that the proteins that resulted were had a fitness deficit. So how do we know if they had a fitness deficit? And so this is going to be one, this is normalized to the wild type. And so this is kind of saying, okay, we're going to compare if the mutation didn't have any effect, we would see that the cells would kind of have their bars be right here. And if you had a fitness deficit, then it would be to the left. So we see that most of these were really well tolerated, the ones that they're showing us here, but some of them were completely not tolerated, and some of them had big fitness deficit. I'm honestly not sure what the asterisks are indicating, and I don't see it in the legend, but yeah. Normally the legend would have all that information. Although they're all only showing us some of them, they also have a table S3 where we can go and see like all the results. They're also kind of highlighting some specific results. And they're saying, okay, well, some of these were well tolerated. And sometimes, too, if you're having trouble interpreting a figure, one of the things to do would be to look at the text and see what the text is telling you. Not just the text in the legend, but the text in the main text in like the results section. And they'll often be highlighting kind of specific things to look for in the figure. For example, they point you to kind of LPXD, which tolerated substitution of all of its isoleucines for valine without having any kind of fitness deficit. And SEC-Y, they talk about how they changed the isoleucines to leucines. It's a little weird that for most of these, they actually only show kind of one of the changes. So they show, like for LPXD, they're showing isoleucine to valine. But they don't show it for isoleucine to leucine. So I assume that you'd have to go to table S3 if you wanted to find that information. But for some of these, they actually do show you both of them, and they indicate that sometimes the results are not the same. And for example, TRMD, which is an essential tRNA methyltransferase, it tolerated isoleucine to valine, but not isoleucine to leucine. And so if we look at this TRMD, we see that isoleucine to valine, that's fine. But if we look down here for TRMD, isoleucine to leucine, now you've got about a 50% fitness deficit. So some of these swaps are kind of like, okay, that's okay. And some of them are like, not so fast. And so then they're kind of, okay, this is showing kind of proof of concept that this can often work. But it's not going to be as simple as just going through and changing all the isoleucines to valines or isoleucines to leucines. We're going to have to do a bit more work. And they're saying 22 of the proteins that they tried to make, they couldn't actually make. They were having problems. They couldn't get viable colonies, and so that could just be that they were killing the cells, and so then they couldn't get any growth. And some of them, they were just having kind of problems with their cloning strategy. And so they were only getting kind of partial recombination. So they were getting these kind of these products that weren't actually what they were trying to get with their recombinating. And so basically things aren't as simple as they might hope. But this does show kind of some proof of concept. But they're going to have to be doing more than just substituting the isoleucines for valines or the isoleucines for leucines. There's a lot of proteins, there's a lot of potential combinations, and there's only so many brain cells. And so let's see how we can maybe use some artificial intelligence to help us out. And so now they're going to be using some different software tools, some different deep learning tools and things in order to try to predict what changes they could make in order to keep the cells growing happily. They talk about how there's a lot of new kind of development in these tools that make it a really good time to try this out. And they're going to try it out using some different tools. And different tools that not only take into account the sequence similarity from evolutionary conservation, but also kind of take into account the structure of the protein, what's nearby in the protein. So not just what's next door in the sequence, but what's actually next door in kind of 3D space. And so we'll see how they're able to take into account both information from the amino acid sequence, like the neighbors in the sequence, as well as the neighbors in kind of 3D space. And they talk about how different kind of modeling programs emphasize one of these things versus another, and how they're then able to combine the strengths of these different programs in order to make different predictions and then compare them to one another and see which are best suited for different purposes, and to mix and match kind of structures that were predicted by these different models in their quest to recode this entire ribosome. So I'm going to actually just kind of go through this figure and kind of illustrate some of the strategies that they're going to be using, and we'll also look at kind of the text. But I think that sometimes a visual representation is helpful. So they take this input protein and they're going to mask the isoleucines. And so what do you say mask? Basically, they're not telling the software that there's kind of like an isoleucine there. They're kind of hiding those positions. And they're saying, okay, software, now tell me what amino acid you would put there. And so they can either just hide the isoleucines, or they can hide the isoleucines as well as kind of neighboring amino acids. So by neighboring, you can talk about in 2D, and so what's nearby in the sequence, or in 3D, so what's nearby not only in the sequence, but actually in 3D space. And then they have these different kind of designs that they're going to make. This I to V, basically here they're just taking every isoleucine and changing it to valine. They're also going to be doing this with like the masking. So this is kind of the strategy we were looking at before, which was just, okay, let's just change this, make this straight change in all these positions. But now they're saying, okay, now let's predict and make changes to any amino acid in those positions. And so we hide the isoleucines and we say, okay, well, tell me what would you put there instead. And that's how we get this kind of like ESM2. They also have this MSATR. And so here it's drawing in evolutionary conservation information from this multiple sequence alignment and saying, okay, well, what would normally be in that position in these other species other than isoleucine? And so let's put that there instead. And that's going to be this kind of like MSATR. And those are relying on this protein sequence. These are these protein language models, these PLMs. They also have these structure-based sequence model. And so here you're going to have that masked sequence, and they're going to take the structure of the protein, and then they're going to try to predict the sequence based on the structure. And then they're also going to have tools that are then going to try to predict the structure based on the sequence, taking into account the evolutionary conservation as well as the kind of structure to sequence information and this like AlphaFold and all this stuff, and get this more complicated model. And they're going to give us a little more information in here. So they're talking about these PLMs, these protein language models. They can learn self-conditional probabilities, so the probability of a sequence, by training on large protein databases. And evolutionary link variations can be further captured with MSA transformer, which uses related sequences to model conditional probabilities. So the probability of a sequence given that alignment would be what you get with this MSATR. With the PLM, it's kind of the probability of a sequence based on training with protein databases. So here it's showing us more about this PLM strategies. And so remember here, we're kind of taking the sequence, we're hiding the isoleucine positions, and we're saying, okay, predict what you would put there instead. They can do this either simultaneously or autoregressively. With the simultaneously, they're saying, okay, we're going to mask them all, and then in one shot, okay, tell us what would you stick in each position. And then that would give you this kind of simultaneous version. With autoregressively, you say, okay, we're going to mask them all. We're going to ask you, okay, change, what would you put here? And now we're going to take the sequence where you have what you had put there. And so now we've replaced one of the isoleucines in the sequence. And now we're saying, okay, next isoleucine position, what would you put here? And so then you're taking the information based on kind of like the sequence the new sequence you had from that first replacement, you're incorporating that and now saying, okay, well, how might that affect what you would choose for this next position? And so they have these kind of like two strategies that they can use for this. And that's what they're discussing here. And then for each of these, they're going to kind of choose the one with the highest probability. So stepwise versus all at once. And they're also then emphasizing too that this MSA TR, so remember MSA, that's multiple sequence alignment, it's going to incorporate additional evolutionary information from the MSA of the target genes orthologs across the entire bacterial kingdom. So this is taking into account the evolutionary conservation of this position in order to say, okay, this one's most likely in evolutionary conservation terms, and so that one might be a better substitution to make than a substitution to another amino acid. Or at least evolution thinks so. But that's just looking at sequence, and so now we're saying, okay, well, sequence is important, yeah, but if you just see an amino acid sequence in a line, that's not really telling you about what's going to be nearby in the 3D structure. And so now they're going to take into account kind of structural information in both the 2D and the 3D. So with the 2D, this is kind of looking at steric interactions of adjacent residues, so sequence neighbors. So remember steric, that's talking about kind of like taking up space. And so if you have steric interactions of adjacent residues, what that's basically saying is that next door neighbor amino acids are kind of getting into each other's space. And so it's kind of your tree's over my fence type of situation, and you don't want that to happen. And so you might want to take into account that what's next door. If you've got like one of those tryptophans next door, one of those big bulky guys, then that kind of might make it so that you don't want to stick a big bulky amino acid in the place of that isoleucine. So that'll be the 2D information, and then they also want to take into account things that are nearby in space in the folded up protein. And so in the folded up protein, you might have interactions between regions that are kind of spatially distant in the sequence, but spatially close in the 3D structure. And so they can look and incorporate the information from the 2D information as well as from the 3D information to get better idea, to hopefully get a better kind of prediction of what to put in that place. Okay, so now they're going to tell us about this other model, protein MPPN, which is going to be this model. And here they're masking the sequence like we're used to, but now they're incorporating the actual protein structure. And so here they're taking the protein structure from the protein data bank, and they're saying what is the most likely amino acid residue in that position based on being able to form a protein that has the structure that we want for this protein. So we're saying, okay, we want the protein to maintain its overall structure. So what amino acid would you therefore put in these places? As opposed to kind of like what would be the most likely amino acid to be in that place, would be like how can we best maintain the structure for this? What could we replace these isoleucine positions for? And so this protein MPNN tool is going to evaluate the probability of a sequence given a structure. And then they're also going to use AlphaFold, which predicts the probability of a structure given a sequence. So with this protein MPNN, you say, okay, we've got this structure, what is the likely sequence? With AlphaFold, you say, we've got this sequence, what is the likely structure? And then they kind of combine all these models together in order to get this kind of uber modeling tool. And so they tell you more about kind of their tools, and there's like this AlphaFold design, which is back propagates through AlphaFold to find sequences matching a target experimental structure. And then they have like this custom extension where they're using then the information from this protein MPNN, as well as they can incorporate information from this kind of protein language models into this final model. And so I'm not a machine learning person, and so I'm not going to try to explain all this, but they have kind of this complicated framework. The bottom line is it's taking into account information from those different tools to try to make the best predictions about what they would want in that place. And if you want more of the methodological information about that, you can go to the methods, and it's going to tell you about kind of all of those different things. And then you can also like kind of go and download code and stuff. One of the main things to know about these methods is that they have kind of temperatures, and when they talk about temperature, that's basically when you think about temperature, that's energy, and so you're allowing the molecules to kind of explore space a little, and rather than getting stuck in a rut. And so if you think about a hill, like this landscape with hills and valleys, if you are stuck at the bottom of a valley, then it's hard to kind of see even that there might be a pot of gold on the other side of the mountain. And so by giving you more energy for you to climb over and see that pot of gold, then you're able to kind of explore a larger range of space. And so similarly, what they can do is they can kind of start with a high temperature where they're allowing these amino acids a lot of variation in what amino acid is placed there, and then kind of narrow down the temperature so that they're saying, okay, you can't change as much at this position. And so to try to keep the model from changing too much and to hone in on those optimal ones. And so that's one of the things that they talk about with this temperature, is kind of going through these cycles of starting at the high temperature and then going to the low temperature. So now they're going to go ahead and they're going to kind of compare their different models. So ultimately they're going to go and they're just going to do this for the ribosome. But first they're going to start by evaluating the different models and how they performed on essential genes. So they want to try to kind of find the optimal one that satisfies both of these models, the structural based and the evolutionary based and things like this. Now they want to go and compare their models. And so they're going to compare models that were used for like evolutionary based methods as well as structural based models. And they're going to compare versions of the 50 essential proteins generated by these different models. So by this ESM2, so one of these protein language models, as well as by one of these structural based models, this protein MPNN. And they're going to see the difference in the amino acid distributions at each position by calculating the Kolek-Leibler divergence score between the probability distributions predicted by the models. Okay, we don't really need to know about how this KL thing works, but we do want to know the key information, which is that it's looking at the divergence between the probability distributions predicted by the models, which quantifies how their substitution preferences diverged. And so we're saying, okay, so at this position, like what is this most likely to be replaced for in these different models? And so here what we're looking at in this C is a scatter plot comparison of ESM2 and protein MPNN residue selections for all isoleucine positions in 15 essential proteins. And they're showing the distribution of entropy for isoleucine substitutions with color indicating the KL divergence between the models. And the sequence logo showing examples of residue preferences at the representative positions. And so basically what they're doing is they're taking the proteins, and they're saying that for the different isoleucine positions, how much kind of flexibility is there in what amino acid is at that position. And so you can see that on the x-axis, we have the distribution entropy for isoleucine residues predicted by this protein MPNN. And then on the y-axis, we have the distribution entropy for isoleucine residues predicted by this ESM2. When we talk about entropy, that's kind of like randomness or disorder. It's like the number of different things you could have at a position, basically, in this case. And so if you have high entropy, that means that you can have a lot of different amino acids in that position. Whereas if you had low entropy, that would say you always see this one amino acid in this position. And so if we're kind of closer to the origin, we have less variability. Farther from the origin, we have more variability. If we are more to the right, that basically means that in this protein MPNN model, we see a lot of this variability. If we're far to the top, it means that with the ESM2 model, we see a lot of variation. And so you can see, like over here, with this ESM, we see almost no variation. We always see isoleucine here in this evolutionary model type thing. But if we have it where we are using this protein-based model, now we see that we can get a lot of, for a structural-based model, now we can see we get a lot of variation. We have high entropy, as opposed to over here where we would have kind of low entropy in this structural prediction, but high entropy in the sequence prediction. And then if over here we have high entropy in both, meaning that there's a lot of flexibility in what amino acid is placed there. So this is kind of just showing the big kind of key takeaway I take from this is that these models often have very different kind of strategies that they alight upon in which structures, what substitutions they make. So remember, they're not just making like a single change and giving you one model, they give you a lot of models. And so they're saying in all these models that they generated, what is the most likely amino acid that they're going to tell you should put in that place where there was an isoleucine. But they allow too that the isoleucine itself is actually put there, and so that's why you see that you do still see isoleucine in these positions. And so some of these are like, yeah, you probably really, the isoleucine there might be really, really hard to change because these software tools are predicting that there's going to be an isoleucine there almost all the time. Whereas over here, okay, well now this is more kind of like loosey-goosey. Go ahead and change it. Nature changes it. Our software says that you can change it in all these different ways and still get a protein that meets the requirements that you want. So what this is highlighting is basically these software tools come to different solutions in different ways, but by looking at them, you can get information about the importance of various residues at various positions, as well as this kind of gives them an idea that, hey, maybe we might actually need to use different software tools for different purposes. And in the future, we'll want to look into seeing how these tools are kind of coming to these different things. And they also talk about how, like, this is all different from the background. So this is like the background distribution of amino acid residues. And so they're saying, like, the ones that predicted by these models are different from the things that we would see just like in natural variation. Okay, so that was kind of evaluating their tools and kind of some strengths, limitations, differences between these tools. And now they're like, okay, we did that with some of the essential genes, and what's next? Let's go ahead and do it with the ribosome. And so we're not at the point where we can replace all of the amino acids, isoleucine amino acid residues in the entire genome, but let's do kind of a proof of principle, and let's do it for the E. coli ribosome. And so this is a big hunk of complex. It's got a lot of different proteins. It's got RNA. It's got a lot of interactions between these protein subunits. So this is going to be a big challenge. The ribosome also makes up a large proportion of all of the protein in the cell. And so if we can do it with the ribosome, then that's going to be a really big proof of concept. So let's look at what we're dealing with. It's the largest protein complex in the cell. 52 essential protein subunits, 3 ribosomal RNAs, and 382 isoleucine residues, with an average of 9 per subunit. And so they're taking us back to figure 1D. And so here we're taking us back here where we see that we have these 382 isoleucine residues in essential proteins of the ribosome. And there are 52 proteins that those are located in. So they've got their work cut out for them. They're also directing us to figure F6. Let's see what's in F6. Now they're showing us the number of isoleucines in each of these proteins. And so you can see, like, we've got an average of about 8 per protein. And here they're showing us kind of the number of mutations that they have to introduce in order to make those changes. So you'll see that sometimes they have to actually introduce more changes than you would have it just like making a one-to-one change, because they have to introduce changes to the neighboring amino acid residues, neighboring in either 2 dimensions, so the next door neighbors, or 3 dimensions, so the people across the street from you. So 2 of the subunits don't have any isoleucine. Phew! These RPMC and these RPMH. But the rest of them, they're going to have to actually go ahead and change. They're going to start by seeing whether nature has already changed that, and that might give them some insight into whether this is feasible and which ones might be easier to change, and whether they can incorporate evolutionary information from those proteins themselves to see, okay, well, maybe we can change this to this amino acid and all would be good because it happens in nature already. And so what they're going to do is they're going to look at the amino acid frequencies in those genes in other bacteria. And so this is what we see in figure S7. So we go to our supplemental, we see figure S7. Here they're showing us for these different genes, so these are the genes of the ribosomal proteins. They're saying this amino acid, it's showing us the kind of the prevalence of species that have that protein without that amino acid, basically. And so they're saying that for these proteins, there's a lot of the bacterial sequences don't have cysteine, say, but very few of them don't have alanines. And then figure B, they're kind of just normalizing that to take into account the length of the gene. And so what we want to be looking at now is this isoleucine, and you can see that there's only a few that actually don't have isoleucine, so this might be a little bit of a challenge. They're not very naturally occurring ones without the isoleucine. But there were some. So 16 of the 52 ribosomal proteins had natural variants where they didn't have isoleucine. And so then they're going to kind of be using those as a comparison in some cases. But they find that the structures that they eventually come to don't have close sequence matches to the ones that were from nature. But although they found those ones in nature, they wanted to try to kind of use them in the lab and see if they really were functional. And they found that not really for some of these. And so although they saw some of these like natural ILE-less ones, these natural variants that don't have isoleucine, they actually had this big fitness deficit compared to the wild type. And so you can see kind of the doubling time. That's how long it takes for the population to double. And so the longer it is, the longer the doubling time, the slower it's growing. And then they're kind of in parentheses are quantifying how it compares to the wild type. So this would be like it only grows 27% as well. And so even though these are kind of existing in nature, when they try to make them into the lab, then they're having some problems. And this is suggesting that they're not, these reported natural variants are non-functional or severely impaired. And so just trying to look at evolution isn't gonna cut it for their redesign. And so how are they gonna go about their redesign? They're gonna start by redesigning each subunit individually. They're gonna test each of those individually, and then they're gonna try to combine things. So they're taking us back to figure 2B to show us kind of like the strategy for doing these predictions. And so they're going to make these predictions, and then they're going to actually go back and do that kind of design, build, test. And so that was the design, and now they're going to build it with this recombinering, so using recombination to replace and engineer kind of these bacteria to have versions of the genes without isoleucine. And then they're going to have different versions of these that they can test. By making these changes in the bacteria, they're then able to just test the fitness deficit. They're able to test the bacterial growth in order to see what effects those changes had on the bacteria's fitness. One of the technical things that they need to kind of account for is they don't want there to be undesired crossover recombination between the variants and the wild type genes. And so basically what they did is they don't want the sequences to be too similar to the wild type, or else you're going to keep getting kind of like swappages and stuff. And so they incorporated synonymous codon substitutions every 2 to 8 nucleotides across the coding region. So what this means basically is they're changing the DNA spelling to change the RNA spelling without changing the amino acid spelling. So they're changing the sequence, the encoding for the protein sequence, without changing the protein sequence that's encoded for. So it's like changing the spelling of gray with an E to gray with an A. And those would be synonymous. They would both kind of indicate the same color, but they would have different spellings. And so they need to do that so that the cells don't accidentally swap kind of one gray for another gray. And this is important to note because the sequence sometimes does actually have an impact, like the underlying sequence, the DNA and the RNA sequence, that can have impacts on the expression of the gene. And that's something that they say that they need to kind of take more into account in future tools when they're trying to incorporate large-scale changes in the genome, is it's important to take into account the underlying sequence information and how that sequence information is used, not just the protein sequence information. And so that's something that is kind of going to be more of a challenge moving forward that they need to start incorporating more. But for now, they have a big enough task at hand. And so what they're doing is they do though need to account for those potential changes by making this kind of control where they take those ribosomal genes and then they have they put in versions with all those synonymous mutations without actually having the ones that make the changes to the amino acid. And so basically they have these synonymous versions where you have the same amino acid specified, but using different spellings. And then they have versions where they have those synonymous mutations and then additional mutations on top of that in order to change the sequence. And so this is just kind of like showing you that there was some fitness deficit for some of these changes that they had to introduce, but overall these cells were okay with you changing the codon spelling. But in the future, you might be able to kind of optimize that using tools that take more into account the underlying sequence. But for now, this is what they're going to be using. So now they're going to test out their different strategies. And they're going to test these different kind of computational tools. The first was just kind of like this brute force that we started with, where we just go and we change every isoleucine to a valine. And so we can see that for some of these, it actually worked fine. So for some of these proteins, they changed from an isoleucine to a valine, and the cells still grew greater than 90% compared to the wild type. Now what they're going to do is they're going to say, okay, well, some of these was still not working. And so we can see that if we change the isoleucine to a valine, some of these didn't grow at all, so we've got the 0. Some of these were like 50%, 0 to 50%, so they have a severe deficit, and some have like a mild deficit that's 50 to 90. So can we do better? And so now we're going to kind of get higher and higher up in terms of how complex our models need to be. And so this ESM2 model that we're going to incorporate, so we kind of like take into account kind of predicting the sequence with one of those protein language models. That worked for some of these. Some of these were still not working, and so now we had to go to that MSATR, which takes in the evolutionary information. That worked for some of these. We now go to protein MPNN, which is going to take kind of the sequence from the structure information. And that is then going to give us some more of these, but some of these were still not working. So now we got to go to our AlphaFold design. Now we're going to still have some problems. We go back, we take that protein language model, and we incorporate the 3D information, so your neighbors, not just next door, but across the street information. That'll give us some of them, and then some of them we still have these fitness deficits, and then we're going to have to go and do some brute force mutagenesis. And so that we're going to see they're going to do some saturation mutagenesis, which basically says, okay, let's go and let's change each isoleucine to a different amino acid and just see what works experimentally, so empirically. What did the results of the growth assays say? So some of these were easy and some of these were hard. And so that's basically what they're just explaining here and here. But I just walked you through it in the figures. And so like I said, sometimes when you've got a lot of practice with reading papers and with the familiarity with things, sometimes it's easier to just go straight to the figures, and that can also be helpful to make sure that you're not being biased by the text itself, and then you could go and read the text itself. But sometimes if you're not really familiar with the method, if you're confused about something, it's good to have to be reading the text along with the figure. But to make it easier, you want to have them kind of like open side by side, or I find it's helpful. So here they're just kind of like summarizing what I just said. And now they want to look into a little more depth. So if we look at 3B, here they're kind of comparing the solutions that they came to with those different models. And so with this ESM2, this is one of those protein language models where it was kind of predicting what would be in the place of the isoleucine. And then this MSATR was drawing in the evolutionary conservation. Protein MPNN was going from structure to sequence, and then the AlphaFold was kind of going from sequence to structure, and then it was also, they're customizing it to kind of bring in information from other tools. What we can see is that it's colored by, so at the bottom you kind of have the amino acids in order of the frequency at which that amino acid was changed, like an isoleucine was changed to this amino acid. And so, for example, like for this one, for the ESM2, the most often change was an isoleucine to a valine, and that was the most common change in all of these. And then it changed, the next most common was to a leucine. Again, that's common with all these, but now you see they start to diverge. And so, for example, you have over here that you're seeing kind of this K and this R, whereas with this model, K and R, like from the protein MPNN, would be very rarely substituted, whereas it's substituted more commonly in this ESM2 model. Bottom line is they're making different types of changes. And so this is interesting because you're seeing these, so this would be like MSATR, this is looking more at the evolutionary stuff. And so just by looking at the residue type, so whether it's nonpolar, polar, acidic, or basic, so the coloring, you can see that the coloring distribution is a lot different with these, which is saying that the evolutionary solutions are not always the same as or they can be very different from the solutions that are generated based on these models. And so they're kind of like exploring different space than you would see with this tool. And you might even be seeing some changes that you really wouldn't expect to see. I was a little confused because they talk about like the MSATR and the ESM2 were selecting polar and branched chain amino acids, but structural guided models frequently favored biochemically distinct ones, especially arginine at a higher rate. Whereas like here, arginine seems to be favored fairly high compared to in the protein MPNN. It's not favored very much. And so I was a little confused by that statement, but for me, the bottom line is that they're coming to these different distributions, and some of these changes would not be things that you would expect. Here they're just kind of talking a little bit more about when they had to get into the more challenging ones. So like this N3D, where they're taking into account the neighbors in 3 dimensions. They're saying, okay, we considered like 3 to 7 angstrom radius, so basically that's the distance of how close they have to be included. And those would then be ones that would kind of be masked in the sequence to be like, okay, now you can change this amino acid, as opposed to just being able to change the isoleucine. But they say they don't want to change beyond 7 angstroms, so they don't want to get too far away, because then you kind of get these long repetitive sequences that are unlikely to be functional. So now they get like a lot of potential structures, and then they actually used AlphaFold 2 in order to help them to kind of rank which ones were most likely. And so they're going to be ranking which of the models that they generated is going to be the most likely to be successful, based on like the different models kind of came to similar conclusions. So what was kind of like the highest probability with both models type of thing. And then this is then going to allow them to choose which ones that they want to actually experimentally validate. So now let's go and actually see the results. And so we can see the results in a couple of different figures. And so like they're going to have a figure here, figure 4, and then they're also going to have figure S10. And they have these kind of similar layouts where they're going to be showing us the different positions, relative positions in one of the proteins, and then these different isoleucines, and then what the isoleucine was changed to in these different design softwares in the sequence, and then the fitness compared to the wild type. So, for example, at this, if we made these changes, so this was like one of the predicted models, and you were changing this isoleucine to an alanine, or you were changing it to a glutamine, or you were changing it to a valine. And so like in this model where they changed it to a valine, then they saw this kind of like mild growth deficit. But again, this is like not just changing that one position, it's changing all of them. And then so that was the I to V, where all of them were just changed to a valine. Here you're kind of like changing some to a valine, some to other amino acids, and then these are just the different models that they tested. And some of these changes are going to be into isoleucine, and some of them are actually going to be, as we'll see, changes in nearby residues, because they were allowing for those changes to take place. And they also found that the changes that were made in these different models were varying greatly from one another. They kind of like highlight some of the substitutions. And so I'm just going to show you a few of the different things that they highlight. And so in the case of this RPLU, so this 50S ribosomal subunit protein L21, the most fit variants were generated by structural models. And so they're saying, now they're directing us to 4A. And so they're saying these structural models were giving us kind of better fitness than these other sequence-based models that we were looking at. This is the sequence logo of the common variants from the MSA. So this is kind of like in nature, you'd find these amino acids. And so you can see that you're changing it to things that would not be found in nature very frequently. And so they're like, okay, well, why are they making those changes? Unfortunately, with these protein language models, you can't really know, because there's a lot of kind of dark box stuff going on. But they wanted to try to kind of like post-rationalize how these changes were made and why they kind of still worked. And so they say, like, okay, well, for this ESM2, it clusters sequences with similar identity. And so because it wasn't favored in nature, then it wouldn't be kind of like in the model, showing up in the model and stuff, and then it would be less likely to be used. And so then you kind of have the neighboring, when you take into account the sequences of like the 3D space interpretations, then you kind of increase the likelihood that that place would be predicted. And then it's kind of further trying to rationalize why they would put an arginine in that place. And they say it's kind of interesting in that this position is kind of on the surface, but even though it's on the surface, you can't just replace it for any old thing. So a lot of time there's kind of like a lot of flexibility at the surface, but not here. And so it's saying that you can kind of take into account incorporate more information into your model in order to better propose solutions possible solutions for the protein that have a better fitness. And so like taking this model and incorporating 3D information into it, say, could then bolster it to the point where it could then come to some of the solutions that are being found by more of these structural-based models. And by comparing the results from these different models, you're able to kind of figure out some more information about structure-function relationships. They also found that sometimes they had to kind of make compensatory mutations elsewhere in the structure in these other amino acid positions. So like this alanine 12, if they made this change to an alanine, then they were able to kind of provide a stabilizing interaction nearby that was then able to compensate for the change to the isoleucine. So I'm not going to go too into depth here, but basically there's structural information that can then allow you to make changes that would stabilize the region around the mutation while still allowing the mutation to take place, even if the mutation alone would be incompatible with life, basically. So A was this RPLU. If we look at this RPSJ protein, here they're saying, okay, this one was trickier. So here even with our like I to V, this simple substitution, we were still like, we got a little bit of a deficit, but it was still growing okay. But if we try to do that with this other protein, well, now these E. coli are screwed. And so with this protein, they found that their simple substitution models just like were not working. Even their AlphaFold design, their protein MPNN, their structure-based methods weren't working very well. And so then they had to go back and kind of fully redesign parts of the protein. And so when they took that ESM2 model, which here did not work at all, basically, and so here was this like protein language model, and they just tried to run it as is, it didn't work. But now when they incorporate the 3D information, they're able to get the bacteria that are basically have normal fitness. And they had to basically, when they looked at the sequence that this protein made, that this software predicted, it actually made a lot of different changes in this whole region of the protein. And so it made this big change rather than just trying to make single changes. And so they're showing here in the structure where these changes were made. So they're like, okay, well, that's weird. We changed this like whole big thing. And how does this compare to what we find in nature? Basically, they try to do this PCA analysis, this principal component analysis, where they group together sequences that are kind of like similar, and they try to cluster them into related things. And what they found was that this sequence was most closely related. It wasn't like very closely related to anything, but it was most similar to bacteria from like whole other species. And so they're like, okay, that's a little weird. This is something that evolutionarily wouldn't be come across probably, but you're kind of with these AI tools, you're able to sample from proteins from all sorts of different species and not restrained to evolving within a single species type of thing. But this didn't work for all of them, and so some of these proteins were still very resistant to having functional versions made. And so what you're looking at here is kind of the pseudo-log likelihood based on evolution. So this is the evolutionary probability of coming to, like, that this protein would be functional type of thing. And then this is the AlphaFold categorical cross entropy, which is a measure of the model confidence. And then you see that the things in triangles are the machine learning designs, and then the circles are the mutagenesis designs. And so basically what they did was, for the ones that they still couldn't get to work, they used saturation mutagenesis, where they went into each isoleucine position and they changed it to every other amino acid. And then they looked to see whether the bacteria grew. And if the bacteria grew, well, you see green, and if they didn't grow, you see this red. And the farther you are to the right, the more kind of like evolutionary probable you are. The farther you are on top, the more likely the AlphaFold model kind of like predicted it, or like the more confidence the AlphaFold tool had. You can see that sometimes both of these were very, very confident, and the bacteria were like, not so fast. And then they had to actually go and use kind of this brute force mutagenesis in order to just go and change them and see what they get. And so that by doing that, go and change them, they were able to eventually come across designs that worked, those green circles, but they weren't necessarily the ones that you would expect based on their hard work tools and evolution. Instead, it's kind of just like, okay, we still got to brute force some of these. As for how they did this brute forcing, so basically they talk about this NNS mutagenesis by multiplex automated genome engineering, or MAGE. Whew, okay, so NSS mutagenesis. Don't know what this is? Okay, go look it up. So NNS, basically what it's saying is that for the N's, you're going to change those positions in the codon to any other nucleotide. And for the last position, that wobble position, you're only going to change it to a C and a G. And that's going to kind of minimize the amount of possible swaps that you can make in terms of the DNA sequence. How they're actually going to make the swaps is then going to be through this MAGE, this multiplex automated genome engineering. And so basically what they do is they take these cells and they put in DNA pieces that have different, that incorporate the mutation. And then they have the cells kind of do that recombination inside of the cells, kind of like we saw before. But here what they're doing is they're actually introducing the mutations, and you kind of just have this pool of DNA that have like all these different mutations to make, and then you put those all into this like pool of bacteria, and then it's kind of like survival of the fittest. The bacteria make the changes, and then the ones with those changes that kind of like have the best fitness are then able to grow the best, and then you keep doing this in cycles, and you keep adding pieces of DNA that then lead to these cells kind of like coming across an optimal solution through these various cycles, and then it's all automated, and so then that's where you get like the A and stuff. And so they did all of this, and they found that they got those results where they were eventually able to come to a functional version of each of those proteins in the E. coli ribosome while conserving the function. But there's a problem because that was all doing it one at a time. And so one at a time is hard enough, and now they're like, okay, well, we did that one at a time, and yes, we did it in the bacteria, but the bacteria, we now need to actually, if we want to do this really make this feasible and be able to say, yeah, we changed all these amino acids in the bacterial ribosome in these cells, we got to change them at the same time. Thankfully, the bacteria make this somewhat easier for us in the fact that a lot of these genes are actually going to be made in the form of, like, from operons. And so basically, a lot of the genes are transcribed and then translated at the same time as part of these kind of like two operons. So these two kind of chunks of DNA that also conveniently are right next to each other. And so if they exchange this whole big piece of DNA in the ribosome, for the modified version, they don't have to go and change each gene individually. However, that's a really big stretch of DNA. And you can use those short pieces of DNA in order to kind of put in smaller changes, like just like linear DNA in order to make smaller recombinations. But when you have a big old bulk piece of DNA like this, how you're going to introduce it is going to be harder, and they're going to actually use this bacterial artificial chromosome. And so think like a really big plasmid type of thing that you stick into these cells, and now they're going to actually use this kind of CRISPR-based method where these cells are then going to, inside these cells, which are then going to be expressing or basically making CRISPR, so the gene editing tool. They give it the sequence that kind of like is neighboring. They guide it to the sequence in the bacterial artificial chromosome, the modified one. What they're going to do is then they're going to have the CRISPR-Cas cut that sequence to cut out that new version of the operon. And then they're going to have these cells then recombine it. And so replace the normal operon with this modified one. Then they were also incorporating an antibiotic resistance gene, and so that's going to then allow them to grow in the presence of the antibiotic, and only the cells that actually took in the modified version are going to be able to grow. And they're going to be able to grow and grow and grow and grow and grow and grow, and then you can measure that fitness, at least in theory. So what they do is they've got these different kind of versions that you'll see that they had to make. And so this RB1, this EC19RB1, they're like, okay, let's swap out the 21 genes that are all part of this thing. And so you can see like some of the genes are actually not in those operons. And so here you're seeing kind of like, there are different ones throughout the genome that they're going to actually have to replace individually, but these are the ones that they can do in this one piece, these like yellow ones. So here you're showing you like the yellow, this is the proteins that are part of those operons. Blue are proteins that are in other places. The red dots are places where they had to do those isoleucine changes. And then the gray is going to be the RNA. So you see they've got their work cut out for them. And when they try to just replace all of that operon, the cells didn't grow. And so they're like, okay, we're going to need to do some more work. Well, first let's make sure that what we actually put in these cells is what we think we put in the cells. And so in B, they're just kind of showing junction PCR. So basically they're saying like, okay, if the changes we made were actually there, we would be able to see that, see a PCR product showing us that if we kind of looked for regions that incorporated overlapping regions that we were looking for, we would be able to see that we actually got a product. But if we didn't actually incorporate those changes, then we wouldn't see this PCR product. So the DNA is not the problem in these cases, but the cells still can't grow. So we've got all these different positions. So which of these proteins is actually causing the problem? And so to figure this out, now they're going to start and they're going to kind of like chop off pieces at the different ends. And so what they do is they kind of go, so this is the 5' and this is the 3' end. They shorten it from the 5' end. So, okay, we just have this portion of the 3' end and the cells grow fine. Okay, it's not one of these genes. Let's lengthen it a little more. Okay, it's still not. The cells are still growing fine. In fact, we can go all the way up until including this part, and all of this is fine. So the problem must lie somewhere over here. We know if we go one further, we kind of go to this RPLW. Booyah, the cells don't grow. Okay, so it seems to be this RPLW, but is it just the RPLW? Now let's start from the other end. And so if we have this short piece with the RPSJ, we're good. We have this one, we incorporate RPLC, we're good. And now we start to see problems with we incorporate the RPLD and the RPLW. And if we just have this piece where we kind of just take out RPLW, it grows normally. So RPLW really seems to be the culprit here. And when they go and they look at the sequence, they realize that this was kind of like this technical complication where they have what they call refactoring. In the gene itself, there's kind of this overlap between this RPLD gene and this RPLW gene. And the start of the sequence with the instructions for RPLW overlaps with the end of the RPLD. And it overlaps basically in a very suboptimal position because the second letter, the second amino acid in the sequence for RPLW is an isoleucine. And so it's one of those ones that's going to need to be changed. And what they did when they were doing the individual protein subunits was they had this refactoring where they basically then moved this sequence to be not overlapping with this sequence. So they put them next door to each other. And then when they made these changes, they made this functional proteins, and it was functional when you incorporated it this way. But when they tried to do that with the other genes, also, now you don't get this functional things made. And so what they realized was that you actually needed that overlap for some sort of regulatory region or something in order for this protein to be made happily. And it's just, indeed, like if they looked at the evolutionary stuff, they actually see that overlap is very common in nature, and they think that that's kind of one of the reasons why they were having this failure when they did it this way. Before they figured that out, though, they also looked at kind of like the protein level. They wanted to see whether the problem could be differences in the interactions between the different proteins, because that was something that wasn't taken into account when they were doing their individual protein engineering, was that these proteins are actually working as part of these complex things. But they found that there kind of wasn't much difference in terms of the isoleucine, the protein-protein interactions between the proteins or within the proteins. So they're like, okay, we don't think that this is really the big thing. So now they needed to find a position that maintained the overlap and maintained the stop codon that you need for RPLD, and so this meant that there were fewer things that they needed to test, and then by brute force testing all of them, they were able to come across this viable solution where they were changing the isoleucine to an asparagine, and then that allowed it to grow. And they want to see, okay, well, is this stable? And so what they do is they basically use ALE, adaptive laboratory evolution, to grow these bacteria and grow these bacteria and grow these bacteria and grow these bacteria for 450 generations. So basically 450 doublings of the cells. And then they're going to go and they're seeing, okay, well, is the fitness of these cells changing over time? And importantly, are these cells picking up additional mutations, and are they really what they care about is most is probably like, are these going back and going backwards from where they were? And so, like, reverting to having the isoleucines in those positions. But what they found was that they actually didn't get those reversions. And so those cells kind of stayed having those isoleucine swappages. The changes that they did see over evolution were for things that they would kind of predict based on adaptations to the artificial growth conditions in the laboratory, rather than changes to overcome deficits from the isoleucine. And so you can see that their growth rates kind of like didn't change as well. Also in this supplemental, we can see kind of the growth rates. So this is kind of like showing you the growth in LB, so lysogeny broth, compared to a less nutrient-rich food, M9, this minimal media. And so you can see that the wild-type E. coli, as well as the E. coli with the kanamycin cassette in them, so the kanamycin that's got the antibiotic resistance, but not the actual change. These all grow better in the richer media, the LB, than the M9. But all of these kind of versions that they're showing here were actually growing, including that RB12, which was that one with all of the 21 genes in that operon recoded. And they find that kind of like the average fitness loss per recoded gene was about 1 to 2%. Now they kind of talk about the discussion, and so we really discussed a lot as we went through. But they're saying, yeah, we did it, woohoo! And it was harder than you might expect. And we proved proof of concept. We see that this is possible. There's still speculation regarding what was kind of in isoleucine's place in these evolutionarily before isoleucine was a thing. But there could be really hard to find out, because there's so many different possibilities that are possible. And with these artificial intelligence tools, we're able to come across possibilities for different sequences that can lead to similar structures that we might not expect those sequences to have, like, be able to form that structure. And so there's still a lot that we're finding out. And these tools, however, do have limitations. And so we saw when we were looking at that one thing, they had to resort to just going and brute force testing every of those combinations in order to find one that would actually work. So we still need this experimental data from these methods, from experimental methods, in order to be able to better inform these computational tools. But we can also take advantage of these computational tools in order to see what sorts of things that they're telling us. Like by looking at differences between different types of models and things, we can figure out information about the structure-function relationship. One of the key things, too, was that there might not, like, in addition to not being one right answer, and there being lots of different answers, there's also a lot of different combinations. And so here it's going to get even more important to have kind of more capability to distinguish which ones are kind of like most likely to be correct, so that you don't have to experimentally validate each of these individual things, like if experimentally test each of these different things. Here's where techniques that are very high multiplex, you can test lots of things at once, and you can rely on kind of natural evolution in the laboratory to do things where kind of cells select for ones that are able to grow. But you also need to take into account, too, the kind of like DNA level things and the RNA level things. And so by focusing just on the protein, you might be missing some of the regulatory information at the DNA level or at the RNA level, as was seen kind of when they had that overlapping region. And so even we saw just by changing to synonymous codons in that one position, you altered the fitness of the cells, which is why they had to use it as kind of, take that as the synonymous recoding as the background, rather than just having the wild type as the background. And so that was what we were kind of here. Also, another key lesson was that you can't just change the one residue. You might need to change neighboring residues, which means making even more changes. So this is a really cool first step, and the challenge is going to be, well, one of the challenges is to really then take this not just to the ribosome, but to other parts of the cell. And then, like, whether it's easier if you kind of like, if you think about all of those changes you need to make throughout the genome, is it then easier to kind of just try to synthesize things from scratch or make the changes in the cells? How are you going to verify all these changes? And so there's a lot of different things that need to go into this, and a lot of thinking, but I think that just kind of exploring this paper, it raises a lot of interesting questions and gives you a lot of insight into protein design and into structure-function relationships. And I also think it's kind of exciting for opening up potential space in the genome. A lot of the things to try to like incorporate additional amino acids, trying to expand the genetic code by having like 4-letter codons and things. But if you could actually just have like, oh, any place, we can actually use these isoleucine codons to incorporate non-canonical amino acids, but do them in different places and then kind of use where it was an isoleucine for a different amino acid. And so, yeah, that's kind of like one of the cool things that could potentially come out of this article. Overall, I totally geeked out about this, and I cannot believe that I just spent like 3 hours talking about it, so I apologize, and I don't even know if anyone's still here. But if you were still here, hope you enjoyed this, and yeah,